Welcome to Cell Biology for Pharmacy. This video is the third video in a series on the cell cycle. In this video, we learn about how eukaryotic cells can control the cell cycle and why it's important to do so. Let's begin. In order to ensure that eukaryotic cells replicate all of their DNA and organelles and divide in an orderly manner, these cells possess a complex mechanism that regulates all of these processes, called the cell cycle control system. This system is a complex molecular mechanism that consists of many proteins, enzymes, and other molecules working together to regulate all cell cycle processes. We will not discuss in detail the molecular aspects of this system, but just to give you an example, here we have one of the enzymes that have an important role in cell cycle regulation, called cyclin-dependent kinase, or CDK. There are several types of CDKs, and their concentrations vary during the different phases of the cell cycle, as you can see here on the left. Back to the cell cycle control system in general, the main function of this system is to guarantee that all of the processes in the cell cycle occur in the correct order, and that each process must be completed before the next one starts. In other words, DNA replication must be completed before G2 starts, and mitosis must be completed before cytokinesis, and so on. This complex regulation is accomplished by a system of feedbacks from processes that are currently being performed. Let's look at an example for a cell starting DNA replication. If the conditions are met, the cell cycle control system will signal to the cell to start replicating DNA. DNA replication occurs during the S phase, and once that is completed, the cell continues to the G2 phase, during which it grows in size. Throughout the S phase and the G2 phase, a feedback system feeds information back to the cell cycle control system, hence the name. The cell cycle control system uses this information to decide on what to do next. If the information it gains says that DNA replication was successful and all preparations for mitosis are complete, it will signal to the cell to enter the mitosis phase. However, if there are any errors during DNA replication, or interruptions or delays, the control system will halt the cell at the G2 phase and not continue to mitosis. This system of feedbacks occur at every phase of the cell cycle and allows the control system to determine when to proceed to the next phase safely. Without this feedback system, any delays or interruptions in any of the cell cycle processes will be disastrous. This feedback system ensures that any delay in a process must also result in a delay in the next process in order to maintain the correct order. In this example, we saw that all DNA must be replicated correctly before mitosis begins. Another example is if DNA is damaged. The cell cycle must be paused at G1, S, or G2 in order to provide time for the cell to repair the DNA damage. In the previous page, we saw that the system of feedbacks provide information to the cell cycle control system in order for it to decide on its next action. The decisions that the control system makes during the cell cycle can be summarized in this figure. There are three main points at which the control system processes information it receives from the feedback system and either provide signals for the cell to continue to the next phase or apply molecular brakes to stop the cell cycle temporarily or even permanently. These brakes are termed checkpoints and mainly occur at 3 points: at the transition between G1 and S, at the transition between G2 and M, and during mitosis. So let's discuss each of these checkpoints in a bit more detail. At the transition between G1 and S, the control system monitors the external environment and also confirms that it is suitable for the cell to perform DNA replication, which is in the S phase. This is an important step because DNA replication and cell division requires sufficient nutrients and specific signal molecules to be present in the external environment. An example of these signal molecules are growth factors, which are molecules produced by other cells that signal to the cell to continue growing and reproducing. At this checkpoint, the system also checks for DNA damage and repairs that damage before entering the S phase, since it would be disastrous if the damaged DNA is not repaired and is replicated in the S phase. Now, if these conditions are not met, the cell will decide to arrest at G1, which means that it will pause the cell cycle or enter a specialized resting state known as G0. We will discuss G-nil in more detail later. In animals, this checkpoint is especially important. The presence of signal molecules produced by other cells stimulate cell proliferation when more cells are needed. Conversely, these molecules are blocked when more cells are not needed. This system of stimulating growth and blocking signal molecules controls the number of cells in the body. Now, you can imagine if this system breaks down, it will lead to uncontrolled cell proliferation, which means cells can reproduce without limits, and this may lead to diseases such as cancer. The second checkpoint is at the transition between the G2 and M phases. At this checkpoint, the cell decides whether to start preparing for mitosis or whether to prolong the G2 phase. The control system checks whether all of the DNA have been replicated properly, and if there were any damage to the DNA, the control checks whether those damages have been repaired. If all is well, and the cell has 2 sets of complete, undamaged DNA, it will prepare to begin mitosis. However, if there were any delays in DNA replication, or the DNA has somehow damaged and is yet to be repaired, the cell will prolong G2 to provide time for the DNA to be repaired. Lastly, during mitosis, the cell faces another checkpoint. We will discuss this in more detail in a following video, but in summary, the control system makes sure that during mitosis, all of the duplicated chromosomes are properly attached to the mitotic spindle, which is a structure that will split the 2 sets of chromosomes apart into 2 daughter cells. This is a critical point because the 2 daughter cells must have the exact same genetic material. Any errors in this process, such as chromosomes not attaching properly to the mitotic spindle, would result in either of the daughter cells not having a complete set of DNA. Here we discuss the checkpoint at the transition from G1 to S, because this checkpoint is an important decision-making point for the cell. So, as we've seen previously, the cell cycle control system constantly monitors the internal and external environments for cell size and extracellular signals. Based on this information, the control system can decide to start preparations for the S phase or decide to delay entering the S phase. Once the cell decides to enter the S phase, the rest of the cell cycle generally completes relatively quickly. In mammals, it takes about 12 to 24 hours to complete the whole cycle. That's why this checkpoint is usually considered as the start of the cell cycle, although it is a cycle and has no actual starting point. However, when the conditions are not right, maybe the cell has not grown to a large enough size or there is DNA damage, cells may decide to delay progress through the cell cycle, to wait until the conditions are suitable or the damage has been repaired. To do this, they can either withdraw from the cell cycle temporarily or permanently. In either case, the molecular mechanisms of the control system are dismantled or taken apart. To withdraw temporarily, cells enter a resting phase called G-nil. In this state, the cell does not reproduce, but they are still able to reassemble the control system quickly when they are ready to continue dividing again. For example, most liver cells are in G-nil, but can be stimulated to divide again if the liver is damaged. To withdraw from the cell cycle permanently, cells can enter a terminally differentiated state in which the cell cycle control system is dismantled completely and irreversibly. Many cells in the human body are terminally differentiated, which has some types of muscle cells and nerve cells. This is why some degenerative diseases occur, because nerve cells cannot reproduce. That's it for this video. In the next video, we will look at mitosis and cytokinesis in more detail. Thank you.